So everybody should have open this worksheet right here. We're just going to break into some smaller groups. Please turn your cameras on so that we know you're in the room. Can anybody remind me how to read this square? In 3 letters like CCC, the first letter we get from the left-hand side, and that will limit us to a row. Okay, so C is going to limit us to this row. The second C, we read from the top. So that's this one. That limits us to this column. So these row and columns come together in this one box. The third C identifies the position within that big box. And this is spelled out, so it's pretty easy. But if it weren't, we would look on the right-hand side. So you go left, top, right. And I would find it's in the second position, so right here. So that's proline. We're going to write P-R-O right there. So how about we each take a different one?
Yeah, it's glycine.
Okay. Or Lee, are you ready with yours?
PHE.
Oh, phenylalanine. And then Jack, did you have AGU?
Yeah, it was like the SER one.
Serine. And then Natasha, UAC.
TYR.
Tyrosine. And Mac, CGU.
I don't know what it is, but the ARG.
ARG is Arginine. And then Emma, you got CCA for us?
Yeah, it's THR.
Rheonine. Lucy, GAA.
GLU.
Glutamine, glutamic acid. Good. Okay. Oh, I don't like that. Didn't match. Let's put that over here. TH. So sometimes you'll get this wheel. So the wheel is you would read the inside to the outside. So like your first letter, so let's take our GAU, right? So the first letter G is going to limit us to this pie piece. A, we go to the second row, so now I'm narrowing my pie piece. And then GAU, and I would end up with the same, it's the same amino acid as you do in the big box. It's just 2 different ways of finding it. Some people find one easier than the other. So it's good to recognize either one. You will have this on the test. I use this square, so be familiar with this square, okay? We're gonna have a test Friday. So now we're gonna do these codons, and Arginine I'll do so that you can see how to do it. Okay, so Arginine, ARG, I might find it easier to do this wheel. Look for Arginine right here, okay? So remember, this is the first letter A, second letter G, and it could be AGG or AGA. So I think the wheel might be easier to work backwards from. So that is GAA or GAG. I'm going to write on this. GAA, GAG. So proline went to Jillian. Any luck with proline, Jillian? So is it AUG and AUC? There should only be 3 letters. So you had proline. Let's go back up here. Proline. Okay, so let's look here. For proline, you would get C, C, and any 4th letter. Let's double check over here. Proline, yeah, CC and any 4th letter. Right here is proline. Again, you can use either box, you'll get the same answers. So for this, you could have CCU, CCA, CCG, and CCC. That's called the wobble effect, the fact that the 4th letter or the 3rd letter can be changed and you often get the same amino acid. Okay, boys, did you get valine? Let's see if you guys agree with each other. Lee, what'd you get?
I had GUU, GUC, GUA, and GUG.
Okay, Jack, did you agree with that?
Yeah, I got the same thing.
Okay, so again, that one, any fourth letter works. The Wobble Effect. Okay, phenylalanine. Did that go to Alyssa and Lucy? No. Who had phenylalanine? I forget. Natasha, did you? Oh, Mac? Okay. What did you come up with for phenylalanine, Mac?
I was a little confused. I only got the first one, and it was like UUC. Maybe.
Good. Yep. So, phenylalanine is right here. So it's UU and then either U or C in the third position. And I can see that in this box here. So UUU or UUC, either one works. Maybe I'll add the wheel in case people find the wheel easier. UUU or UUC. Okay. Oh, I see. Histidine. That's what you had, isn't it, Alyssa, Lucy? Yeah, see, they shifted. That's why I didn't know. Okay, Alyssa, what'd you get? And then Lucy, you see if you got the same thing.
CAU and CAC.
CAU, CAC. Do you agree with that, Lucy?
Yep.
All right, I will trust you guys. Since two of you. Glycine, we had Natasha, I believe.
GGU, GGC, GGA, and GGG.
Okay, good. The box shows us GG, anything else. Good job. Okay, so both of those are skills that you will use on Thursday's test. So speak now or forever hold your peace. Do you guys have any questions on how to do that? No, Ghost Rider. Okay, so the three letters come from, do they come from mRNA or tRNA? Which one am I reading up here? mRNA or tRNA? What does it say on the left? It says first base and codon. So is that referring to mRNA or tRNA? It is, thank you. tRNA is the anticodon. So it's really important when you're doing your test that you recognize you're reading the message, right? And that makes sense. It doesn't make sense to read the anti or the transfer. It would make sense to read a message. Okay, so we're reading messenger RNA. Okay, what is the start codon? So looking up there, you should find methionine. And methionine is the start. So what three letters indicate the beginning of a gene sequence? All genes will begin with this three letters. What you got? What do you think, Emma? What three letters indicate the start? Start or methionine, M-E-T. Either one works. They're the same. We can see a start and methionine here. We can see a start and methionine here. What three letters?
Oh, sorry. AUG.
Yes. Okay, so the AUG is the start. And there's three different ones that code for stops. Matt, can you tell me one of the stop codons?
UAA.
All right. And Natasha, can you give me another one?
Where should I look again?
The stop codon, you'll see over here on the wheel, skinny ones, or you'll see here in the orange. So the stop codons, and we already have UAA. So what's another one?
Can one be UCA?
So UCA is going to be something else.
Oh, so UAG.
UAG. And there's one more. And what is that third one, Jillian? We have UAA and UAG. What's the third stop codon? What do you got? UGA is correct. Okay. So the boxes are fairly easy. You just have to recognize where to look, what to look for, huh? Okay, so now this is your original DNA sequence. We want to transcribe it into mRNA. So complementary base pairing, for example, T will bond to A. A will no longer bond to T because T doesn't exist in mRNA. Instead, you have a U, right? Uracil, and G. Okay, so I'm going to transcribe into mRNA, and then I'm going to read the amino acids. So that's where I'm going up to the decoder box or wheel. And AUG is methionine. We've already found that one. So then here I'm going to write methionine, MET. Okay. So we have 3 of those to do. AUG is star. AUG is also methionine. Jack says yes. He's the second one. So Jack, how about you give me yours, and Lee, you let me know if you agree. All right.
Okay, so I got AAG, UUA, CCA, GAU, and CCC.
Okay, you
Yeah,
agree
I
with
agree.
all of that, Lee?
Yep.
Okay, Lee, what do you got for the amino acids? And then Jack, you can tell me if you agree.
It's methionine.
AAA,
It's LEU, or I don't know all the words, the full words. LEU.
leucine. Yeah, you don't need to know the words. It's fine. So leucine,
PRO.
proline,
Aspirin, or and stop. Or no, wait. No, I was looking at the wrong thing.
no, CCC shouldn't be a stop.
No, yeah, I was looking at the wrong thing. CCC. Pro, yeah.
Okay, Jack, do you agree with those? He was under pressure, so did he get him what you got?
I got most of all of them, but like the first one, like instead of LEU, the first one, like AAG, is LYS. Like leave the LEU because there's 5 of them there, but there's only like 4 that you wrote down.
Oh, I missed the first one. So lysine, and then was it LEU next?
Yeah.
Okay. Okay, girls. Lucy and Alyssa, are you girls ready? Okay, Lucy, you want to give me the letters?
Yeah. I got AUG, GUA, CCU, UAA, UVA.
What was the last 3? UGA?
Yeah.
I don't have it in front of me. Okay, and then Alyssa, do you agree with those?
Yeah.
Thank you. And then can you tell me the amino acids? And Lucy, you double check the work, okay?
Start, val, pro, stop, stop.
2 stops in a row.
Mhm.
Okay, Lucy, is that what you got?
Yep.
Great. Okay, Natasha, can you give me the mRNA strand? And Mac, check the work.
UGU, AAA, GUC, UGG, CAG.
Hey, Mac, you agree with that?
Yes.
So, Emma, I can guess that you have the amino acid sequence, and Natasha can check your work. So what you got? Okay, so UGU, we're gonna go U across, G is here, GU, UGU, cysteine, CYS.
Oh yeah.
AAA.
LYS.
Mm-hmm. And GUC. Yep, valine. And UGG. That's tryptophan. And then CAG.
CHR.
CAG?
Yeah.
GLN.
Oh, sorry. GLN.
Yeah. Okay. Okay. So from RNA, you should be able to find the DNA. From RNA, you should be able to find the tRNA. From the tRNA, you should be able to find the amino acids. So you want to be able to work, given any of these pieces, you want to be able to work above or below it. You guys have any questions on that one? Skills we'll do Thursday. Okay, here's our last one. So we did a very similar picture. So this is protein synthesis, correct? It's actually the Central Dogma because it's the whole thing. So number one, what do we think number one is? It has 2 strands. Shout it out, be brave.
DNA.
Yes, that's right. So what is DNA gonna make? When you transcribe it, you make number 2. And specifically, I can tell from its structure what type of RNA.
Is it mRNA?
It sure is. Whoa, look at that. And then this is all taking place in number 3. What is the structure? Give me a female voice. Number 3. What is this whole structure inside of the cell?
Cytoplasm.
Oh, look, it's surrounded by a nuclear membrane.
Nucleus.
Yes, thank you for being brave. Okay, so outside of the nucleus, number 4 is going to be our cytoplasm, the open space. So I know this is Central Dogma. I know this is the nucleus and this is the cytoplasm. So transcription occurred inside, right? Translation occurs outside. So now I got to figure out what these different structures are. What do you think number 5 is? Remember we had something with 2 subunits, a large and a small. I can see the mRNA went out of the nucleus and lined up here at this snowman looking thing. What is this snowman looking thing? What do you think, Lee? The site of protein synthesis.
Ribosome.
Yes, that is correct. Okay, so mRNA is lined up on the ribosome. Every 3 letters on mRNA is a what, Natasha? Every 3 letters on mRNA. We were just decoding the box with 3-letter codes. What do you call those 3 letters?
Those codons?
They sure are. Okay, and opposite of that, we find 3 letters that are complementary. What is opposite the codon? Jillian. Opposite codon. Binds to. So AAA versus UUU. So you have a codon and then you have a blank codon, meaning opposite. Blank codon. Okay. Do you see I'm trying to think of some English words. You have a protagonist and a what?
Oh, anticodon?
Yes. It wasn't quite a good example, but I was giving you, but I was trying to get there. Okay. So the 3 letters opposite are the anticodon. And what do I call the whole structure that carries the anticodon? We often describe it as a clover leaf. This structure here, what do we call that? Emma? What carries the anticodon, or where do I find the anticodon? Number 8 is an example of one of these pieces. We're down to 3 words: proteins, amino acids, and tRNA.
tRNA.
Correct. So we typically describe that as a clover leaf. This drawing doesn't really have it as a clover leaf, but the tRNA, this thing here, On one end, we find the anticodon, and what do we find on the other end? What is it carrying? Natasha, what does it carry? tRNA is bringing individual what to the ribosomes? Building blocks of proteins.
Amino acids.
Correct. And then Lucy, I see we have added a bunch of these circles, each being an amino acid. We've added a bunch of them together to make number 10. So what do you call a bunch of amino acids connected together?
A protein.
Correct. Good job. So trying to walk you through like how you would think that out, right? So images help us make sense of information. Okay. Any questions on that or how to figure out what is what?